We're going to continue talking about how cell membranes allow things in and out in this lecture today. We talked about this idea of passive diffusion, right? Passive transport. And what that means is particles are going to move from high concentration to low concentration, which is called the concentration gradient, and it doesn't use any energy, right? That's what passive means. Passive, if you're a passive person, you just kind of sit back and relax. You let things happen, right? So passive diffusion is this idea that particles, if they're concentrated, really tightly packed, if there's a lot of them in one area, particles move, they randomly move, they're going to push each other away to an area where there's less of them. And that makes sense when you think about it, right? If you're packed into a room with 100 people, you want to get out of there. You want to move to an area where there's less. And particles do the same thing. They go from high concentration to low concentration, and because it's just a random movement, it doesn't require any energy. Well, today we're going to talk about a different type of passive transport called osmosis. And it works under the same idea of going from high to low concentration, but osmosis is very unique in the sense that it talks about one specific type of molecule, and that is water, okay? So when you hear the word osmosis, the first thing you should think is water, okay? That's it. Osmosis, water. And why are we talking about water as being its own special thing? Why is there a term for water and it's not just passive transport? It's because water is so important for life, and a lot of times it actually travels the opposite direction as other particles. So let's get started talking about it. So what is osmosis? Well, as I just said, it's the diffusion of water across a selectively permeable membrane. So water oftentimes is going to move through a membrane where other particles can't, because water is pretty small, right? H2O is not a large molecule. So a lot of times water particles or water molecules can get through an opening in a membrane that a larger molecule could not. So if you take a look here, what you see is osmosis at work. These red circles represent some type of solute, right? So in this case, it's sugar molecules. And the blue dots here represent water molecules. And what you'll notice is at the beginning of this little demonstration here, the water level on both sides is equal to each other. And if you take a look here, on the left side, we have a whole bunch of red circles, not as many blue. So there's more solute than solvent. And on the other side, we have a lot more solvent than solute. There's only 3 of the red circles as there are the blue circles, right? So what would happen in this scenario? Well, if we just left it alone, based off of what we talked about in the last lecture, these red circles would go to this side, right? They would want to go from where there's high concentration to where there's low concentration. But they can't. Why can't they? Because they're too big, right? If I want to try to get a piano through a doorway, I can't, right? I have to take the piano apart to get it through. Well, the same thing is true with these molecules. They're just too stinking big to get through the openings in this semi-permeable membrane. So what happens? Well, what happens is that water is going to move to equalize it. So if you take a look here, after a certain amount of time, the water molecules are small enough to get through. So there's more water molecules over here, so there's a higher concentration of water on this side, so the water is going to go this direction. The sugar wants to go this direction, but it can't because it's too big, so it can't go. So what ends up happening is the water molecules move over to this side and the water level rises. So they're not the same height anymore. The sugar can't go anywhere because it's too big, right? So osmosis is the same idea as diffusion, except we're just talking about water. And the reason why a lot of times water can travel is because it's so small. We got to go over some terms now that we're going to be using a lot for the remainder of this unit. So water moves across a membrane until both sides are equal to each other. Now we used a term in the last lecture called equilibrium, and it's the same idea, but because we're talking about osmosis and water, we're going to use a different term. It's going to be isotonic, okay? Isotonic. Now an isotonic solution means that there's equal amounts of water and solute. Okay, so the solvent is the water and the solute is going to be whatever we dissolve in the water. It could be salt, it could be sugar, it could be anything that is polar and water soluble. And that's an isotonic solution. Well, what if they're not equal? What if there's more of one or more of the other? Well, there's some terms for that as well. Surprise, surprise. Let's go through them. The first one is hypertonic. Hyper means more, right? So if somebody's a hyper person, they have a lot of energy, right? If I have a student in my class that's really hyper, that means that they're probably going to be jumping out of their seat all the time. So hyper means more, right? Hyperbaric is, more pressure. Hypertonic means more In this case, it's going to have more solute. So hypertonic solution means there's more solute than water. There's going to be a concentrated solution. So if I go back to my iced tea example, that is going to be iced tea that is way too sweet. There's a perfect recipe for iced tea where there's the perfect amount of iced tea mix and the perfect amount of water, and it's delicious. If there's too much sugar, it's too sweet and it's disgusting, right? That would be a hypertonic solution. It's going to be a concentrated solution. Hypo means less than. If you've ever heard of hypothermia before, that is when your body temperature drops too low. So thermal means temperature, hypo means less. So hypotonic means there's less solute than water. So that's going to be a dilute concentration. So what does that mean? That would be very weak iced tea, where you don't have enough mix in there and it just tastes like weak ice. Ugh, it's gross, right? It just tastes like brown water. Who wants to drink that? And that's going to be hypotonic. So I just want to show you a couple diagrams here to explain it a little more in depth. In this little drawing here, I have 3 cups of iced tea mix that I made. And they're varying degrees of excellence, right? So let's take a look at what I have here. What I've drawn is we have blue dots and red dots. The blue dots represent water, and if you remember, water is the universal solvent, so it's always going to be the solvent. And then the red dots represent the iced tea, which is going to be the solute, because it dissolves into the solvent. So let's take a look at my 3 examples here. If you take a look at this first one, I only have 3 red dots and a whole bunch of blue dots. So this is going to be extremely weak iced tea. Why is it weak? Because there's more water than there is sugar, so it's going to be very dilute. And that means there's going to be more water than solute, so more solvent than solute. That is going to be what we call hypotonic. Hypo means less, so it means less solute than solvent. It doesn't mean less water, and that's where a lot of people make a mistake. They see hypo and they remember less and they go, oh, less water. It's not less water. It's less solute. So it's actually more water. So the first one is going to be weak. It's going to have more water than sugar, and that is going to be what we consider to be a hypotonic solution. My next example is going to be really strong iced tea. So take a look here. All these red dots represent the sugar or the iced tea mix, and the blue dots represent water. And what you end up with is an extremely concentrated solution. This is a hypertonic solution because there's more solute than solvent. So there's more sugar than water. I have a way to remember that. Sugar makes you hyper. So if you have more sugar, you get really hyper. So hypertonic means more sugar. Now, it doesn't mean it's always going to be sugar. Hypertonic can be a salt solution, or it could be something else entirely. But just that might help you remember it because sugar makes you hyper. So a hypertonic solution has more solute than solvent. And then finally, we have our perfect cup of iced tea where we have equal sugar and equal water. There's equal solute to solvent, so that's going to be isotonic. Iso means equal, so isotonic means the solute and the solvent are going to be equal to each other. And you know, that to me is a perfect cup of iced tea mix. So both of these samples up here, the weak one and the strong one, are going to be either hyper or hypo, and they want to become isotonic. So things move towards an equilibrium, but in the case of osmosis, we say isotonic. Okay. So we have our isotonic and we have our hypotonic. So this is just another diagram to just show how osmosis works. Water is always going to go from where there's more to where there's less. So water is going to always go from a hypotonic environment to a hypertonic environment. Because remember, hypotonic means less solute, more solvent, which is water. So in this scenario here, there's equal parts water. This is isotonic. If I add some larger molecules here, well, now this side becomes hypertonic because there's more solute than solvent. This side is hypotonic. So what ends up happening is the water goes from where there's more water to where there's less water. So the water is going to travel across the membrane to the other side. We're going to finish up here with one more quick concept, which is called facilitated diffusion. It's the last type of passive transport that we're going to talk about. So far, everything we've talked about is particles going from high concentration to low concentration. But sometimes the particles can't fit through the membrane, so they need a little help. And that's where facilitated diffusion comes in. You might get these proteins. Integral proteins, we talked about this before, right? Integral proteins go through the entire membrane. They allow some substances to pass through more than others. So that would be called facilitated diffusion. it still follows the concentration gradient. It's still going from high concentration to low concentration, so it doesn't require energy, but what you end up with is basically, the way I like to think of it, is a bouncer at the door, right? Doorman. If you ever go to a club, there's usually somebody at the door that lets people in, right? So think of the proteins as the bouncers. They open up and they let particles in. They let some particles in, the ones that can get through. So facilitated diffusion is still passive transport, but it does require a protein to move the particles through. So this diagram just shows that, right? So this would be simple diffusion here. The particles go right through the membrane. They're small enough to fit right through. But in facilitated diffusion, they need to use a protein to get through. So they're still going from high to low concentration, right? There's more outside than inside, so they're still going to follow the rules of simple diffusion, the concentration gradient from high concentration to low concentration, but they need a protein to do it. So that's just the big difference there. So what's the difference between normal diffusion, simple diffusion, and facilitated diffusion? The difference is that facilitated diffusion needs a little help, and that's what facilitate means, right? If I say to facilitate something, it means to help it along. So facilitated diffusion uses a protein, but it still doesn't need any energy. It's still passive. Thank you very much.